get started. Thank all of you for coming out on kind of this cool, rainy Tucson evening for the third and final of the LPL Public Lecture Series for the fall. I'm Mark Marley, the director of the Lunar and Planetary Lab here at the U of A. And every year we have a lecture series that tries to capture some different aspect of the things that we do here at LPL. And this year, our theme is Space on Earth, trying to convey how we study various aspects about space, such as glaciers on Mars, by studying geologic processes or other processes here on Earth. And so our final process for this lecture series is understanding something about life. So before I introduce our speaker, I'll just remind, if you're a student, you're here maybe for extra credit. At the end of the evening, you can get, at the end of the lecture, you can go out and get a stamp from the folks out front that'll demonstrate that you are here. And also we'll do at the end a quick drawing for some of the LPL swag that I'm sure kind of captures the variety of all the things that we do. So with all of those, and the folks online, if you have questions at the end, feel free to go ahead and type those into the chat, and we'll read those to the speaker at the end. So our speaker tonight is Dr. Solange Duhamel. She's an associate professor in the Department of Molecular and Cellular Biology here at the University of Arizona, and she also has a joint appointment here in the Lunar and Planetary Lab. Solange received her PhD in Marine Environmental Sciences from the University of Aix-Marseille and joined university about 6 years ago, came to the U of A. Solange is an aquatic microbiologist and biogeochemist specializing in studying the abundance, diversity, and activity of aquatic microbes in the ocean. And as she will explain more to you, is that's a study of microbes, the ocean, and other extreme environments that led her to the study of microbes colonizing fresh lava flows that we're going to hear about tonight. So with that background, I'll turn it over to Professor Duhamel.
Thank you. Can you hear me well? Perfect. All right, so yes, tonight we are going to speak a bit about multiple colonization of lava flows and the interaction of lava flows with water. So I will give you a bit of background on that. I want to start by explaining why that might be an important topic to study. And so this is an important topic to study for three fields, for ecology, for the study of the origin of life, and for the study of astrobiology. So in terms of ecology, we are interested in what we call primary successions. And so essentially, we are trying to understand how an ecosystem develops starting from bare rocks. And so, I should have tested that. Okay. So any kind of ecosystem needs to start somewhere. And so we think here about a bare system where there is no life. So a sterile environment, bare rocks that are going to be colonized by what we call pioneer species. And those pioneer species have to be microbial, so unicellular forms of life, and mostly prokaryotic, that is bacterial and archaeal. And this process helps what we call the weathering of the rock and transforming it into a soil that can sustain a more complex ecosystem containing vegetation, very simple at first, and then developing into something more complex. So it has to start somewhere again. And so we are really interested in what's happening between bare rock and a very simple ecosystem dominated by microorganisms at first. This is different from the ecological process that is called secondary succession, in which you can have something similar that happens, a bare substrate that becomes colonized by microorganisms and then more complex forms of life. But the difference is that when you have the difference is that in the secondary succession, you have a disturbance that is going to reset an ecosystem. So you actually start with a community that's its climax. A disturbance such as a fire is going to be erase all forms of life, and the ecosystem need to redevelop. But oftentimes this system doesn't start with the bare rock. It still has some organic matter and it's already advanced, so the process goes faster. So this is a distinction. Okay. So here you see a lava flow, and you see basically molten lava rock, right? And so the temperature is about 2100 degrees Fahrenheit. So you can imagine that it's very harsh for life, and it's a sterile environment. There is nothing that can survive such temperature. So we start with the sterile ecosystem, sterile system. It's not an eco. So a sterile substrate, and any kind of microorganisms that is going to be deposited on this substrate is going to have to deal with very harsh environment. That is not enough nutrients. There is no organic matter present. There is also a wide range of temperature variability with the exchange with the air, and there is also low water availability. So what we can learn from studying this ecosystem is learn about what we call extremophiles, so those organisms that are adapted to harsh environments. And because we learn about those extremophiles, we can learn something about early life on Earth and astrobiology. Early life on Earth, because early Earth was very actively volcanic. And so we can get insight into what the early forms of life might have been in terms of colonization. And that brings us then to the application to astrobiology. So in terms of application to astrobiology, the more direct ones are application to Mars. These lava flows are good analog for environments that are volcanically active or have been volcanically active in the past, such as Mars. And it helps us to understand how life might have been present on Mars and perhaps even preserved, so we can study things such as biosignature that we could potentially recover by analyzing samples from Mars. Okay, so with that, I hope I convince you that this type of study is important for different fields. But we are going obviously here to focus on application to Mars. And so here, it's unfortunate because I have all my titles under the Zoom thing. I don't know if...
Yes, I think we can.
Thank you very much. Okay, so here we have a schematic of the geological evolution of Mars over time, and we have timeline of the top here. So those are the Martian time periods in billions of years. And we have all sort of geological processes on Mars. And I want to draw your attention here to the surface hydrogeology and to the surface and atmosphere evolution. So basically, Mars has been going through different phases, and early Mars was warmer and wetter. But there is still evidence of the formation of pluvial systems on the surface of Mars through different type of evidence. Some of them are geomorphological. So, for example, here we have a comparison of the Colorado River Canyon on Earth, and it's compared to the morphology of such type of canyon on the surface of Mars, which indicates that there was likely water digging these channels. And those evidence show that it's been going even throughout an extensive part of the Amazonian. So we have water, and we also have evidence of volcanic activity. So here, very intense at the beginning of Mars formation, and then sporadic volcanism up to the recent geological timescales. So on Mars, volcanic activity could have interacted with the presence of water at the surface at times. So that brings us to a potential development of ecosystems here, because we know on Earth that when lava interacts with water, we get life that flourishes, and I will show you some examples of that. It's also important for the origin of life, because we know on Earth that some of the theories are associated with hydrothermal systems. So where you have a volcanic chamber that is going to warm up some water, you get also gradients, chemical gradients that are very favorable to prebiotic chemistry and the formation of early life. On Mars, there are evidence of such type of hydrothermal activity. So, for example, in the form of these white nodular opaline silica deposits, and also proofs that water may have interacted with lava in the formation of what is called a rootless cone. So this is represented here on this image on the right. So those really look like volcanic crater, but there is no magma chamber feeding them under. They are the results of explosion when lava is meeting cold water. And we will see again why it's important in our research interest. So, okay, so if we sort of resume that, there is the possibility of an origin of life on early Mars. And so if microbial life had been had developed and then find a way to survive for extensive period of time, or at least being preserved in the form of resistance form of life, such as spores, there is a possibility that dormant microorganisms could take advantage of. And so this is something that we hypothesized, and we made some sort of a conceptual model here that I will guide you through. And so basically what you do here is you start with a topography that is confining, so a low topography, where you have ground ice with sedimentary substrate on top of it. And so if you have a new lava flow, it will tend to want to fill this low topographic area. And like I explained earlier, when lava interacts with any kind of water or ice, it's going to create those rootless cones, so those explosions. And we can imagine a system where these lava flow will overlay one another over geological timescales. So why it might be important for biology is illustrated on this slide. So here we have one of those rootless cones. So say we are looking at this little guy here. We have the formation of a rootless cone. We have ice underneath, and we have those microorganisms, those really little green blobs that are nicely preserved. And so you can imagine we are in the scenario where we have another lava flow that is going to warm up the ice underneath where the microorganisms are. The water is going to melt, and the microorganisms are going to be driven closer to the surface. And so that gives us an interesting target to go sample, because you will have to drill less deeply than you would in other environments where those microorganisms may be buried under a deep layer of sediment, basically. Okay, so still following me with that? All right. Okay, so that brings us to the first case study that I want to present to you. So this is the study of an ephemeral hydrothermal system. So ephemeral because it's not long-lasting, at Holuhraun in Iceland. So this is Europe and Greenland, and we have Iceland sandwiched in between. And so zoom in on the islands where we have a huge glacier here, and this little red blob is the lava flow from the Holuhraun eruption. And you can see that it overlays with this huge glacier river that is being formed by the melting of this huge glacier. So when the eruption started, Dr. Christopher Hamilton from Wakefield was very exciting because he's a volcanologist, and so went to study the volcanic eruption and discovered this interaction between the lava flow and the river. So we can see the river here, and so this interaction is creating a lot of evaporation here of the glacial river because there is basically the water is boiling right away. And so when they went after the eruption was over and the lava was that is in the summer of 2015, they found some green stuff. And so they ended up being in touch with me about that. And for disclosure, Christopher is my husband. So that's why we were talking about it. And so, you know, I like to say that life is like a box of chocolate. I like the quote because I'm an oceanographer. So what would I do about microorganisms in volcanic environments? Well, now I'm all about that. So I was really curious about what this green stuff might be. And so I went there. And it was all new to me. I'm used to work on a ship, and at this time I was working on land, but still crossing water. And so this is not a ship. This is a four-wheel drive. We had to cross this river a few times before getting close enough to the site. And so we end up having to camp close by and to set up a lab. So a lot of the results that I will show you have been acquired in that little yellow tent. Okay, so the first step for us was to describe the interaction between the lava flow and the glacial river. And so this is described in this paper. Again, we have Iceland, and here you have the outline of the lava flow. So you can see that it covered part of the river. So basically the river continued to flow through the lava flow, through the rock when it was solidifying, but also underneath. But we also can see that the river, well, at least the melting of the glacier was creating a branch of the river on its side. So this is an aerial image of the lava flow with the source right here, and our river water that flew and underneath the lava flow appeared here. And so what we did here is by thermal measurements, we were able to identify the part of the river that was heated by the lava flow, so it's color-coded in purple, and the part of the river that was cold, which is color-coded in blue. So you can see that between 2015, when they went and saw the green color very intense, there was very little interaction with the colder branch of the river. When I went in 2016, the colder branch of the river was, but we still had a decent ephemeral hydrothermal system in there. So what I wanted to focus on was to characterize the so measure things like water flow, which was in the previous paper, or the chemistry of the water, the pollution, the turbidity. So today I will have time only to present results about biological parameters, but I want to address some of those. Okay, so here we have a representation of the hydrothermal system as it looked like when I visited in 2016. So this is an aerial image with the number of the station that I was able to sample. And here it's a representation that is color-coded with the pink being the warmed water, and in blue, the glacial water that was much colder. And so because the system was not only being fed by the glacial river, we also took samples from a cold spring at Svarta, which surfaced here more not like a lake, but pond basically. Otherwise, it was more like disrupted little pools like this, but it's the same system. So basically, I sampled it here. The water just feeds the system on that end, so on the north end of the hot spring. And you will see how that was contributing some microbial diversity to the hydrothermal system. So the first thing that we did is we thought, okay, we are in a system where there is a lava flow warming water. We have a hydrothermal system. So we should find some organisms that deal with hydrothermal very well. And so we try to isolate them in culture by growing these unreached culture at 50 degrees Celsius. And so doing this in the tent is not ideal. We could probably have done more than that. But we ended up being successful with 11 strains, which is actually already pretty good. And we found that, so then we sequenced them. And we found that most of them, so all the ones that I circled in red here, were geobacillus. And they were very similar. So there is no doubt that was a 100% similarity. And we found one species of Penibacillus. So also pretty clear that that must be the right strain. And those are very well characterized. They are pretty abundant in the natural environment, even in those that are not necessarily high temperature, because they tend to form spores. So they form spores, and when the temperature is what they like, they just start growing again. We also found one pseudo-rhodoplane with 96% similarity, so that sounds pretty good. But when looking at the literature, the previous species that have been characterized cannot grow above 35 degrees Celsius. And so here we have a plot of growth. So this is absorbance, so basically a cell abundance versus time. And you can see the growth of our geobacillus and our penibacillus, and our pseudo-rhodoplane is actually doing pretty well in between. And so we probably have isolated a nouvelle taxa of this order. It would need to be further characterized, but this is interesting. So the thing that we need to know here is that it is thought that about 1% of organisms can be cultured. It's just very difficult to culture microorganisms. So if you want to know what are the microorganisms present in a system, you need to use on sequencing. So you will use omics to figure out the identity of those microorganisms. And so this is what we've done. And so we focused on a gene that will give us an answer, a good answer only for bacteria and archaea. And we ended up finding something like 96% of bacteria. There were very few archaea. So I'm focusing on those. And I will show you those results.
I've never been excited about a small pile of metal flakes before.
The phylum level. So if you think about the tree of life, you can go to different degree of precision. And so the higher degree of precision is the species, but we can go at a higher level that will be less precise, that is the phylum. And I chose to represent it that way because there was already quite a bit of diversity at this level. And then I will present you the result for the species level, but with a different format. So basically here we look at the relative abundance of those species when they are categorized at the phylum level, and different color are going to be different phylum. We have our Svarta cold spring here. So this was in this location. And the sample number 8 is our cold river. In between, we have our samples from the hydrothermal system. And so what you can see already just, by the color code is that there are new colors that appear in the lava heated system. So basically we have new phylums And, that means a lot of different species that appear in the lava heated system that do not exist in the other end members. We also see that some of those exist in the Sparta system at the phylum level, but I will show you later that at the species level they were different. And so basically we have this increase in taxonomic diversity, and we identified some members that are known to be extremophiles, including those thermophiles that we had isolated in culture. So now we are going to show results at the species level, but you can only imagine that if I present a plot like this with color code, it's going to be crazy, the number of colors that you need, right? So I'm presenting the results different way. It's called a Venn diagram. So those circles have different numbers when they are allocated to the ecosystem in question. So here the lava heated system, we see that we have 94 species that are unique to the lava heated system. 6 to Sparta and 7 to the glacial system. Then when there is an overlap, it's how many species are in common between these different systems. So for example, there were only 2 species that were found in the glacial river that were also found in the lava heated system. And so on. So one shared here, only 2 between Sparta and the lava heated system, and altogether we shared 4 species. So what that means is that we have an ecosystem that is created and that looks very unique. And so the question is a bit, where does microorganisms come from? And this is something that we were not completely able to resolve here because we didn't sample all the possible ecosystems that could contribute to the hydrothermal system. I will show you in the next study that we did make this effort because we learned from our experience. However, what you can do is still look at those sequences and compare to what is known in the literature or public database of microorganisms, who they are and what is their and what we found is that many of the groups that were important in this higher diversity in the lava heated system were very similar to isolate in ecosystems that are deep basaltic aquifer. So an indication that those microorganisms that end up in the hydrothermal system must have been brought by the interaction of the lava and the water. And so in my little brain, I see it that way. So that's my very artistic schematic of how I imagine things. But so basically you would see things across. So it's a cross section. And so you have the lava flow that meets the river. So when it meets the river, it pushes the river underneath, but also through it. So as the lava flow down, it's porous and the water can go through. And by pushing down inside the sand sheet that was running over those microorganisms that may have been dormant and just hanging in there, were untrained by the water that was warmed up and ending up in the hydrothermal system where we sampled them and found them to be more abundant. So somewhere along the way, they got reanimated and started to grow, and being more abundant, we were able to sequence them. Okay, so again, this is relevant in the context of astrobiology. So I told you earlier that they are different for lava-water interaction or not. And this image of potential lava-water interaction on rocks is very comparable to our system in Iceland, where you see the margin of a lava flow and evidence for something that could have been digged by the flow of water. And so altogether, this study showed that this is a very interesting environment to sample, and we already kind of knew that, right? We sent rovers to study environment that we knew were aqueous at some point. But here it shows us that if this lava-water interaction happened and stimulated the growth and diversity of microorganisms, those could have been preserved again in the sediment and would be definitely better target than elsewhere in the environment. And going back to our little schematic, it verifies that this idea of microorganisms being more, closer to the surface where we have evidence of water-lava interaction would be an ideal target for future sampling. Okay, so that was the first case scenario I wanted to discuss, where we have lava interacting with water. The second case scenario is going to be a bit shorter. So here we are still in Iceland, and it's an eruption that happened in 2021 at the Reykjanes Peninsula. And so our first interest with this study was this idea of studying primary succession. So we have a new lava flow, and we want to know what are the first microorganisms that colonize this lava flow. So the idea of pioneer species. And so this is work that is led by Nathan Hadlon, a LPL graduate student in my group, and that of Christopher Hamilton. So we've been thinking about the fact that Mars could have had punctuated habitability because of this existence of sporadic volcanism until quite geologically recently. And so the idea was to start by thinking about how a young volcanic terrain could be an ideal place for this primary succession that would then allow the ecosystem to develop. The idea also that, and is represented by this schematic, that lava flow provide new habitats for colonization, and they provide new habitats by the fact that the rock can have different shape and different mineral composition. So different type of microorganisms may be well suited to colonize one type of lava flow or another. So the remaining question, based on this exercise of reviewing the literature, is where to figure out the temporal dynamic of colonizing the lava flow. And also, where do they come from? So just like I said earlier, when you identify the microorganisms in the ecosystem, it's not necessarily easy to figure out where they come from. If it's a very established ecosystem, they come from there. They have been used to this habitat. But when they are new, they don't come out of nowhere. It's not spontaneous like. So this topic has been, since we started working on this, identified as a top science priority in the Planetary Science and Astrobiology Decadal Survey. So this is really encouraging. Okay, so in 2021, we were in Iceland for a sabbatical. And at the University of Iceland, at the University of Iceland, close to the Reykjanes Peninsula. And we started experiencing a pretty intense earthquake, both in terms of frequency and also intensity. And so the intensity went up the roof at some point with a tremor identified with a signal, oftentimes potential volcanic eruption. So knowing that, we hurried to sample a hydrothermal system nearby, as well as soil in the location we thought the volcanic eruption would happen. We also hurried to put in some proposals to the NSF and the University of Arizona to motivate the sampling of this eruption if it would happen. And luckily for us, it did happen. So the eruption started in March 19, 2021, and what started as a little crater became a huge lava flow. So because of its location, it was also close to a populated area and drew the attention of the media, including National Geographic, that highlighted the work that we were doing. So we were really honored by that. Okay, so here the eruption again began on March 19, 2021, and I'm showing you the outline of the flow over time. And as discussed earlier, lava would tend to fill low topographic areas. So you can see how it started to fill up this valley, the Fagradalsfjall Valley. So that's why the eruption is named that way. So we were able to sample the Fagradalsfjall eruption from March to September, and it ended in September. So the whole time the eruption was active, we were able to have bi-weekly sampling. So that includes samples of rock that were very freshly unplaced. So as long as they were not hot enough to go through our sampling bag, we would be able to. And so we first also established station 1, 2, 3, and 4, and unfortunately at some point the lava flow resurfaced this sampling area. So you will see in the data set that we have a lot more data at the beginning for those stations, and then obviously we could not go back. And so we established sites A, B, C that were coming from the same fissure, so they were the same age, so at least we can continue having a logical temporal variability here. We went back a year later to get, the time series plus 1 year. And, oh yeah, I forgot to say that. We used aerial mapping to track the age of the lava. So that's why we knew that these guys were the same age, but also that was allowing to make sure that we were accurate with all our temporal variability. So yeah, a year later, go back to sample, and believe it or not, another eruption starts just a few days after we arrived. So we jumped on the opportunity, obviously, to sample that eruption as well, and go back to our old sampling sites. Another year later, to get a plus 2 years of Fagradalsfjall, we went back, believe it or not, another eruption. So we have a repetition now in Iceland, and we control the volcanic activity. So we went back that year and sampled deeply further, resampled everything else. We went back the following. It was an eruption, but a little bit further away, so we stopped, getting crazy about sampling everything. It's not even on that map. So at the end of the day, here we have 2 years of sampling here, 3 and 4 years of sampling here. So all in one, it's a pretty unique sample suite. There is nothing that exists in the literature that is comparable to that. We have what we call day one lava, but like I said, as fresh as possible. We have at least those bi-weekly sampling during the Fagradalsfjall eruption, but we also have 3 different eruptions, and basically it gives us a triplicate of a natural phenomena, which is also very rare. We also have reference from old lava because this is a volcanic island. So this volcano in particular erupted many times. And so in the area, we were able also to identify lava flow that were aged either 2,000 or 12,500 years old. So that allows us to get a sense of where the ecosystem is hanging to at this location. So again, this is all results from graduate student Nathan Adland. So he did a lot of DNA extraction to get to the identity of those microorganisms. Did also some cell count by microscopy and some screening of microbial metadata. I will not be able to speak about that today, but I will try to speak about most of those first results. But let's start with the cell counts. So here we have on the y-axis cell counts, and is represented as a function of the level of age. And so basically what Nathan did is use a hammer and a pestle and mortar and crush as much lava rock as he could to be able to extract those microorganisms. There is a little bit more, involved steps than that, but the hard work is to do the crushing. And so why he needed to crush so much? It's because the microbial abundance was very low, and so he needed more biomass to try to see something like this. With some background, so you need to prepare a lot of blanks to know how the substrate itself might be fluorescent, and to be able to recognize cells that really have incorporated your dye. So here is a dye that binds to the DNA. So that's how you know that you get a cell. So with those results, again, we have a reference point that is our old lava flow that is 2,000 to 12,000 year oldish. And you can see that we have something that is 10 to the 8, 10 to the 9 cells per gram. And here are all the samples that have been collected. So the ones that are less than 1 year old, you can see that they are very low. It increased over time. Here is a bit of an outlier because it was sampled close to a fumarole. And for reference also, I give you here the range of cell abundance that we find in the Atacama Desert. And the Atacama Desert is thought to be a very good analog for Mars as well because of low biomass due to harsh conditions. And so here you can see that most of this data is either in the low range or even lower, especially at the beginning of the eruption, so in the first 5 months or so. So that means that the sequencing was also a challenge. Obviously, we had to prepare all sort of blanks and checks to make sure that we were not sequencing something that comes from contamination. So once you remove all this potential contamination, we got some results. And Nathan is very good with bioinformatics and statistical analysis and did phenomenal work with all that. So here is a list of all this type of analysis that he was doing. I will not be able to speak about all of them, some of them because I don't completely understand. So the machine learning is the one that I will skip for sure. So anyway, let's speak about what we understand here. I will skip the description of the microbial composition, with all these little colors that I was showing you in the hydrothermal system. We will start by thinking about how the community was composed and how it changed over time. So it's called beta diversity. And so it's also a different way of representing the data here. So we are using what is called a coordinate analysis of the diversity. So he used what is called weighted unifrac. And so basically our goal is to see how similar or dissimilar the different samples are, right? So we want to be able to separate them statistically. So this is what he did, and the statistical analysis takes into consideration the phylogenetic relationships, so how evolutionary related the microbes are. So it's based on their this amplicon sequencing results, and also the relative abundance. So that's why it's called weighted. So how much of each microbe species are used. And so here I'm going to show you the results on this white space. There are two axes that don't really mean anything, so it's a statistical tool. What matters is try to see how the data clusters. And we will have different symbols for the different eruptions and different color for the age of the lava rock. So here are the results. And you can see that already what jumps to the eyes is that it's really the lava rock age that seems to matter in terms of clustering the data, because these signs that represent different eruptions are mixed together. And using the statistical analysis, rather than just our eyes, here are the results. It's clustered indeed by time, with all the samples less than 100 days clustering together, the ones that are about a year clustering together, and the ones that are 2 to 3 year old
clustering together.
So we were able to identify those three groups. So now the next analysis is basically using this PCA plot, but we will be comparing data points based on the temporal variability. So follow me. So here we have the unifrag distance to the previous time point. So we are going to compare the microbial composition of a sample at one time point to the next time point it's going to be sampled at. And then as a function of that, if you get higher values, that means that they are less similar. And if you have lower values, then they are less. Okay. So, for example, let's compare those two. They were sampled at the very beginning of the eruption, and what we find is that they have this value of unifrag distance. Compare it to the next sampling, the unifrag distance value tends to decrease. So that means that those samples, the microbial community in those two samples is more similar. But after the first winter, when we went back, they became more dissimilar, so less similar here because the value increased. Going back just a few days later, it decreased because, again, there is not much time in between, so the microbial community didn't really evolve in the time. We go back a year later, it stabilized. Another year later, it's even more stable. So really what sounds to be happening is that the first winter is selecting for the microbial community composition, and it's stabilizing after that. And so what we found is similar trends with the different sites. What we find is that we have more value, obviously, at all these sites that were surfaced, but the overall trend seems okay, so then there is this question of where do these microorganisms might be coming from? And so what we did here is use a tool that's called source tracking. And to do that, what you need is to have sequence that come from other systems that may have contributed to the microbial community of interest. So how can microbes get there, right? They can get there through deposition by rainwater. They can come back also, they can arrive through the air coming from old lava rock, from the soil nearby. You can see here that there is soil, and we could even see that at the margin, some soil was being deposited on the lava rock. And obviously bioaerosols, so all the microbes that are in the air. So we sample all these different sources. They may not be the only sources, but they are, the obvious one that we can think about. And so here is the result of a statistical tool that is called Source Tracker. And so it uses Bayesian inference model, but basically it compares the sequence from your reference ecosystem to that of the others. So here the aerosol are in blue, the old lava in green, rain purple, soil is pink, and then the unknown is gray. So you can see that here is the source fraction versus lava rock age, and we stop after
the end of the eruption, so
the first 5 months. And so you can see that there is a bit of variability. There is a source that is unknown, but overall, it sounds like the soil and the aerosol are the colors that pop up the most. They are the source that seem to be contributing the most microorganisms to our lab. And so if you continue looking at this analysis after the first winter, it was a complete change. So this time it's mostly rainwater. You can see the dominance now of this purple color. And so we thought it was a bit weird, but the thing is that we obtained exactly the same trend when we sampled the two other eruptions. So it sounds like after the first winter, not only the microbial community sort of stabilize. Change is less than at the beginning, but it's also more similar to what may be provided by the rainwater. Okay, so some conclusions. So again, with relevance to Mars, mostly because of its volcanic activity. So I hope I convinced you that newly emplaced lava flows on Earth facilitate the establishment of microbes. We saw that in the lava-induced hydrothermal system at Holuhraun, the microbial communities from the subsurface environment were untrained to the surface, and they were stimulated by the higher temperature. We found also some microorganisms that were thermophilic. And at Fagradalsfjall, we found diverse extremotolerant microbial communities. So I didn't show that exactly with this dataset, but know that same thing. Most of the sequences, especially at the very beginning of the eruption, were extremotolerant or extremopide. And they seem to be sourced also from soil and the atmosphere before the first winter. So the long-lasting establishment of these taxa after the first winter seems to depend on their capacity to adapt to the environment. So microbes were deposited, made it work for a little while, but then the environment selected them, and the ones that were adapted to those conditions continued to grow. And with that, I will thank you as well as my team and collaborators. Thank you very much.
Thank you, Solange, for that preview of how we might want to eventually go look for life on Mars, which this idea that things are coming from rainwater and the life kind of scatters. Does that imply that maybe you want to look more at places where it's rained or closer to hydrothermal systems? Or if you were to go, have your own rover on Mars to go explore, where would you want to go first?
That's a good question. So with the study at Fagradalsfjall, I think I would relate it more to the ecology of early Mars in that sense. If you need to have a microbiome that would be tricky. But with the presence of hydrothermal system and the possibility that a new lava flow melts some ground ice, I think that would be but also those systems where I was showing you the outline of the lava flow and potential for hydrothermal system, that would be a good place. Those rootless cones, obviously, since we hypothesize that those microorganisms can be pushed closer to the surface, we don't have the means to drill very deeply with rovers, so we want to target places where microorganisms might be closer to the surface.
Interesting. Okay, do we have any questions from the audience? In the back?
Yeah, well, pretty cool study. Nice system. Do you have your top 10 species for the climax community? Do you have like a climax community that you can name, like maybe top 3 species of bacteria? Or can you say that there is a climax community over there?
That's a good question. Well, there is a joke about vegetal ecosystem in Iceland is that if you're lost in a forest, you just stand, you will find your way, because there is nothing really developed to that point where you would find a forest, so in terms of ecological succession, the climax is thought to be when you have something that looks more like a forest or a fully developed ecosystem. Iceland is young enough that you don't find that. There's been some deforestation during the Viking times, but as of today, you would not find that climax community. A lot of the lava flows are covered by mosses. So that would be your reference point to some level, and this is similar to what we sampled at what we call old lava, where mostly lichens, a little bit of mosses. So that was the most developed ecosystem in that region. Sense?
Do we have any questions online from online, maybe?
No questions online.
All right, well, throw it back to the audience. Any more? Right here, go ahead.
So in the first case study, you showed us that you found 11 high temperature species that grow at around 50 degrees. So in the subsequent measurements, did you still find these high temperature species, or was it just that
Oh, you mean
winter?
the sequencing of the sample?
Yeah.
Yeah.
Did winter destroy all of them and just kept
Oh.
the low temperature species?
Okay, because that's two different studies, right?
Yeah.
At Holuhraun, you mean the species that I was able to grow in culture? I found them at Fagradalsfjall. I mean, Geobacillus for sure. The other ones, I don't remember off the top of my head. But they are so common also with the hydrothermal system being nearby. There we found them. They would be very present in Icelanders.
Were all of your samples taken right at the surface, or did you do any cores?
Yeah, no, for the lava flow, they weren't. So we use a hammer, and we were just because they are so low biomass, and we thought the first one would be completely sterile. What we needed to do is be very careful about the way we sample them. So we had special sterile bags, and so basically put them really close to where we would hammer in. And sterilize the hammer before using it. So there was no way to fall. But it's interesting because we submitted a proposal to continue working on this topic. We are not necessarily going to fall that deeply, but in the idea of vertical distribution of microorganisms, we want to do some cross section of rocks that would be probably that size, and even for microscopy that becomes a pretty tedious work. So the idea is that we think that most microorganisms are deposited in the surface, but in terms of finding their habitat, they probably have to go deeper. And it's an advantage for them also to find refuge from those harsh conditions I was mentioning, such as temperature fluctuation when you're at the very surface. And it's probably one of the factors that's selected for those organisms that can survive past the first winter in Iceland.
Did you measure how much the rock had cooled from winter to winter to winter?
Yeah, so it's a good question. Because we were using a probe that we were not bringing to the deep, it equilibrated with the surface pretty quickly. But the residual heat in the whole lava flow would last much longer.
Thank you.
Yeah.
All right. Any more questions from inside? All right. Well, let's thank our
I
speaker
have one
again.
more question online.
Oh, we do have one online. Go ahead.
Yeah. This is a general question. Do extremophiles have the ability to easily do mutations to adapt changes, meaning the difference between generations, that are meaningful to adapt to volcanic events, or is its lifespan doesn't allow mutation during events like an eruption for the species that came from older lava?
I'm not sure I understood the whole question. So the question is about if the microorganisms deposited in the new lava flow from the old lava could mutate to adapt to the new lava flow? Something like that?
Yes.
So that's a good question. Mutation can happen, but not that quickly. Because here we are talking about just a year, basically, not even, because the last time point that we get is in September, and we went back in July, something like that. I don't believe that mutation takes much longer than that to happen. So I would say no. I can't really provide a very thorough
Response on this one, but
the the rate of mutation is lower than the timeline that we are discussing here.
Thank you. Anything else online?
No more questions online.
All right. Well, that concludes again. Thank you very much to our speaker.